Hello and welcome everyone to Bio-Rad's webinar series on Droplet Digital PCR. This is our fourth webinar in a series of 6 Bio-Rad is hosting on ddPCR. My name is Mohini Patil and I'm the Global Product Manager for Droplet Digital PCR at Bio-Rad and your host for today's webinar. First, let's discuss the webinar logistics. The webinar will last approximately 1 hour. Dr. Nick Heredia will present on ddPCR and its application for validation of next-generation sequencing libraries for about 45 minutes. Following the presentation, we will have 15 minutes of live questions and answers with Nick. Throughout the webinar, feel free to use the Q&A box on the left-hand side of your screen in order to submit your ddPCR questions directly to us. We will get to as many of your questions as we have time for. However, if we cannot get to your question today, we will personally follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. Now, a little bit about our speaker, Dr. Nicholas Heredia. Dr. Heredia earned his PhD in microbiology, immunology, and molecular genetics from the University of California, Los Angeles. Nick completed his postdoctoral studies in biophysics at Lawrence Livermore National Laboratory. Dr. Heredia is currently a staff scientist at Bio-Rad's Digital Biology Center, focusing on applications for NGS as well as advanced R&D. The title of Dr. Heredia's talk this morning is Droplet Digital PCR for Next-Generation Sequencing Library Validation. Without further ado, let's get started. Nick?
Thank you, Mohini. Hello, and thank you for joining us. My name is Nicholas Heredia, and I'm a staff scientist within the Applications Group at Bio-Rad's Digital Biology Center in Pleasanton, California. My current focus at the Digital Biology Center is applying Droplet Digital PCR technology on next-generation sequencing. Today, I would like to discuss the following items: a brief overview of Bio-Rad's Droplet Digital PCR technology, a review of Illumina TruSeq and Ion Torrent next-generation sequencing workflows. I'd like to share results of Droplet Digital PCR used to quantitate Illumina TruSeq and Ion Torrent libraries. I will discuss how Droplet Digital PCR can help cross-validate NGS results. And finally, I'd like to share information on how Droplet Digital PCR users can apply the technology on targeted enrichment of amplicons for library construction. These are the critical benefits of Droplet Digital PCR. There's absolute quantification, so there's no need for a standard curve, as you have in qPCR. And this is basically because the number of targets in a given volume is counted directly in each droplet, and this is adjusted for multiple targets occupying a droplet by Poisson statistics. Precision. This is the ability to consistently make the same measurement on replicates. Reproducibility. So this is the ability to obtain the same measurement on the same sample again and again, or from laboratory to laboratory. And sensitivity. The ability to measure very few copies of a target with high precision. And accuracy. The ability to make the correct measurement. Droplet Digital PCR, or ddPCR, is a revolutionary new technology that allows for precise and accurate quantification of nucleic acids. There are essentially 3 simple steps prior to receiving quantitative results. First, a standard 20 microliter PCR reaction containing sample, master mix, and assays, which of course have primer and probe, are partitioned into 20,000 uniform nanoliter-sized droplets by the droplet generator instrument. Second, the droplets are transferred to a 96-well plate, and this plate is thermocycled to endpoint, and that's usually about 40 cycles of PCR. Third, the thermocycled plate is placed on the droplet reader instrument, where the droplets are counted individually from each well by fluorescence to provide a positive or negative result. This is essentially a yes-no answer for each droplet. The data is then displayed in Quantisoft software as a concentration for each 20 microliter reaction, and the units of that are copies per microliter. Here is Bio-Rad's QX200 Droplet Digital PCR system. It comprises of two instruments, the droplet generator and the droplet reader. Bio-Rad offers all the necessary reagents to perform digital PCR in droplets. The droplet generator uses a disposable DG8 cartridge shown in the lower right-hand corner to simultaneously partition 8 independent 20 microliter reactions into 20,000 droplets. All of this occurs in about 2 minutes. The droplet reader instrument can read a full 96-well plate in about 2.5 hours. This system gives the user great flexibility by allowing for a broad range of low and high sample throughput. By allowing the user to decide how many samples to run, from 1 to several hundred samples can be analyzed in a single workday. This is a conceptual slide to illustrate how typically one 20 microliter reaction can be partitioned into approximately 20,000 droplets, each of which give discrete positive or negative yes-no fluorescent results. In real-time PCR, shown on the left, this single reaction tube would only be equal to one measurement, whereas in digital PCR, on the right, there are tens of thousands of measurements per sample. These discrete measurements enable excellent precision and accuracy without the need for a standard reference curve. This diagram demonstrates the power of partitioning a sample into droplets and how this increases the abundance of the target of interest and reduces competition with other PCR targets. The scenario presented here is the detection of a rare mutant target, such as a cancer target, that differs from the wild-type majority by only a single nucleotide. This is also known as a SNP. In this example, there are 40,000 wild-type copies to 40 mutant copies, thus giving the mutant target abundance of 0.1% in this sample. On the left is an example of what would happen in a conventional real-time PCR reaction where all species are present, both wild-type and mutant. Real-time can really only detect the mutant target at levels higher than 1% due to the competition between all the wild-type species. In droplet PCR, however, the partitioning of the sample into 20,000 droplets, basically almost all droplets will have on average about 2 wild-type templates, and approximately 40 of the droplets will have 1 copy of the mutant and also 2 copies of the wild-type. This now changes the mutant target abundance in those droplets containing a mutant template to about 33%, which is easily discriminable from the wild-type. Because of the reduced competition within that droplet from the wild-type sequences, you can basically get much improved detection of the mutant species in that droplet. Due to the Poisson distribution, the occupancy of these droplets will vary in terms of how many exact templates are in each droplet. And of course, our software, Quantisoft, takes into account the Poisson distribution in the results. After that brief introduction on how droplet digital PCR works, I'm moving on to the next section of my talk, the overview of next-generation sequencing. The two current major players in NGS are Illumina and Ion Torrent. They both use sequencing by synthesis method to obtain data. Generally, the user obtains some form of purified sample, either amplicon DNA, genomic DNA, or cDNA. They process the sample to add universal adapters, and at this point can employ barcode adapters if they want to combine multiple samples together. They then hybridize the adapted DNA onto a surface. In the case of Illumina, it is a flow cell, and with Ion Torrent, it is on a nanoparticle. The sequencing workflow then clonally amplifies the adapted DNA on these surfaces. Illumina uses a bridge amplification technique, and Ion Torrent uses an emulsion PCR technique. This clonal amplification is not sequencing yet. It is intended to boost signal detection during the actual sequencing phase. Finally, once the adapted DNA is clonally amplified, sequencing by synthesis occurs. In the Illumina workflow, fluorescently labeled nucleotides are sequentially flowed over the surface. In the Ion Torrent workflow, native nucleotides, or non-fluorescent nucleotides, are sequentially flowed over immobilized nanoparticles in individual wells, and a change in pH is read in each well. This is a generalized overview of NGS workflows and shows that PCR is an essential component. The DNA is fragmented or amplified in cancer panels and then adapted with unique adapters. These adapted library molecules are then amplified by PCR, and the libraries are enriched for correctly adapted molecules. This is the point where accurate quantification can be used, such as Droplet Digital PCR, prior to surface loading. After surface loading, there is another PCR expansion step, which is essentially the clonal amplification on the sequencing surface, the flow cell or the nanoparticle that I described in the previous slide. And finally, sequencing is performed. The question is, how do we know that the NGS sequencing results are quantitative? This is a great point for Droplet PCR, again, to be used, this time to cross-validate the sequencing results. This slide illustrates the bridge amplification method used by Illumina TruSeq. Essentially, complementary oligos to two unique ends of the adapter molecule are arrayed on a flow cell surface. The library adapted DNA is introduced to the flow cell at a low concentration such that one adapted molecule can bind to a complementary oligo and be clonally amplified to generate a cluster. That cluster is spatially distinct from other adapted molecule clusters. The clonal amplification is achieved by synthesizing a complementary strand of the captured target in one direction, which is then able to bend over or bridge to the other complementary oligo. After multiple rounds of amplification, clonal copies are obtained in a cluster around the original single molecule. The purpose of the cluster generation is to boost the fluorescent signal during sequencing by having many copies in the cluster. This is an illustration of how clonal amplification occurs in the Ion Torrent workflow. Nanoparticles, called ion sphere particles, or ISPs, have oligos arrayed on their surface which are complementary to the adapter sequence of the library. The library is placed by the user on the one-touch system at a specified low concentration in order to achieve, on average, one adapted library molecule per ISP. Then, the one-touch system, which is shown at the top in the center, creates an emulsion around the ISPs with an average of one ISP per emulsion droplet. And that cartoon is shown on the right with the circles surrounding the ISP. The emulsions are then thermocycled for clonal amplification and broken open to harvest the ISPs. Downstream of this workflow, the ISPs are enriched for those containing template amplified DNA. This slide is intended to give a relative feel for the cost of running NGS experiments and the throughput which can be achieved by both the lower and high-end sequencer instruments. These costs can change due to the competitive nature of the NGS market, but this table gives a feel for the number of sequencing reads per run and how the runs generally cost between $1,000 and $20,000, depending on the read throughput. Here is an example of how these NGS technologies and associated costs might apply in a clinical setting. Many times, DNA is sequenced from a cancer panel of assays for specific targets of interest. In this example, there are 200 targets or amplicons to be sequenced. A major issue for a single sample is that adding too little library-adapted DNA can miss several mutations, since cancer mutations are often rare targets, while overloading the adapted library can produce mixed reads because the sequencing surfaces are no longer clonal. Often, multiple patient samples would be barcoded and pulled together in the same sequencing run to save time and money. This pooling can cause an additional issue if unequal concentrations of the different pooled patient libraries are present. This would result in one or more samples dominating in the sequencing run and could potentially give false negatives for some patients because the sequencing depth was not sufficient to find the rare mutations due to the unequal concentration distribution in the pooled samples. This obviously impacts researcher time and money, and the runs may have to be repeated due to these issues. This is where Droplet Digital PCR can help with extremely accurate library concentration determinations prior to the loading of the library on the sequencing run. I will now describe the Illumina TruSeq library quantification using ddPCR. The Illumina workflow uses a unique Y adapter shown in the upper left panel, which contains unique sequences called P5 and P7. After the adapters are ligated to both ends of the molecule and the adapted molecule is amplified, the library molecules should have a single P5 and P7 on opposite ends. The ddPCR assay is a two-color assay with one probe targeting the P5 region and the other probe targeting the P7 region of the adapters. The ddPCR assay is circled in green in the upper right corner of this slide. A well-formed library molecule is one which possesses only one P5 and one P7 sequence on opposite ends of the molecule. In the lower right is a two-dimensional plot of ddPCR data. This is plotting all the droplets read in a well with respect to the fluorescent amplitudes for each droplet read. The x-axis plots increasing hex amplitudes, and the y-axis plots the FAM amplitudes for each droplet. The lowest amplitude droplets are the negative droplets, and the higher amplitude droplets are the positive for the TaqMan assays. Thus, in a two-dimensional amplitude plot of the Illumina assay in QuantiSoft, well-formed library molecules will lie in a 45-degree angle from the negative droplet cluster, since these droplets will be positive for both FAM and hex simultaneously. This is shown in the plot at the right with well-formed molecules in orange and the negative droplets in gray. The ddPCR Illumina TruSeq library quantification assay was tested across a thermal gradient from 55 to 65 degrees Celsius to ensure that the assay is robust. The plot on the left is of the concentrations for both FAM and hex channels and shows remarkable consistency across the temperature gradient tested. The 2D plots of the fluorescent amplitude, again FAM on the y-axis and hex amplitudes on the x-axis, show good separation of positive and negative droplets across the thermal gradient. Thus, this is a very robust assay. A unique feature of Droplet Digital PCR is the ability to distinguish the size of the library insert in the two-dimensional plots. Because of this, a probe-based assay has an inverse relationship between the size of the amplicon and the fluorescent amplitude. This is due to the longer the amplicon in base pairs, the less efficient the PCR reaction in the droplet, and thus the lower the amplitude. The highest population in this plot is a cluster of droplets containing only adapter-adapter dimers, as these are the smallest amplicon size and thus the most efficient PCR reaction in droplets. The Quantisoft software is capable of calculating the concentration of the adapter-adapter dimer population by using the lasso tool feature in the software. This ability to quantitate adapter-adapter dimers is simply not possible using real-time PCR. This chart shows in greater detail the well-formed library molecules in orange at a 45-degree angle to the empty negative droplets shown in gray. Extraneous, ill-formed library molecules are highlighted in the red circles and are not at a 45-degree angle from the negative cluster. These likely arise from molecules having only one P5 or P7 sequence, or potentially multiples of P5 or P7 sequences. Again, the lasso tool in Quantisoft can be used to calculate the concentration of these ill-formed clusters, and if significant, could indicate a poor library preparation, which should be remade prior to sequencing. So this really is a new feature which allows you to assess the quality of the library as well. The upper chart is a plot of FAM and hex concentrations of a serial library dilution spanning from 10 to the -6 to 10 to the -9. This demonstrates the excellent reproducibility of quantification across 4 orders of magnitude with the ddPCR assay. The lower chart is all 4 dilutions overlaid as a fluorescent amplitude plot. This is an example of the excellent balancing which can be achieved using ddPCR Library Quantification Kit for Illumina TruSeq. Here, 12 libraries were barcoded and pulled together on a single sequencing run. As can be seen from the blue theoretical dotted line, all of the libraries are very close in terms of the number of reads achieved of each library on the MySeq instrument, which is plotted on the y-axis. Also, the cluster density was about 1 million clusters per millimeter squared, which is excellent and is a strong indication that the concentration of the libraries loaded onto the surface were very accurate by ddPCR. This is the product offering from Bio-Rad for the ddPCR Library Quantification Kit for Illumina TruSeq, which was used to generate the data I just showed you in the previous slides. This is available for sale now from our website. Now I'd like to introduce library quantification for ion torrent using Droplet Digital PCR. Ion torrent libraries typically consist of target amplicon panels, RNA-seq, or fragmented genomic DNA. All of these essentially use the same types of adapters, which can also incorporate barcodes. On the bottom left workflow, circled in green, is a cartoon of the ddPCR probe-based assay, which is a single-color FAM assay and is designed to one side of the adapted insert library molecule. Here is a one-dimensional fluorescent amplitude plot of the single-color probe assay used in the ddPCR library quantification kit for ion torrent. Even though it is a single-color FAM assay, similar types of information about the library quality can be inferred from the one-dimensional plot. Again, like the Illumina library quantification kit, the highest fluorescence amplitude droplets are those containing adapter-adapter ligations with no library insert. This is because they are the most efficiently amplified PCR reaction in the droplets. Because this kit is also a probe-based assay, library inserts have an inverse fluorescent amplitude with respect to the size of the insert. As can be seen in the middle cluster of droplets, those with the highest amplitude are smaller compared to those with the lowest amplitude. This can provide a qualitative analysis of the size distribution of the library. We believe that the lowest positive droplets below the well-formed library insert cluster, but above the negative droplets, represents droplets containing very large inserts, and also ill-formed, potentially chimeric, insert-insert ligations. Chimeric insert-insert ligations would be an artifact of the sample preparation and would be undesirable in a sequencing run. Here, digital PCR can quantitate the number of well-formed library molecules, adapter-adapter ligations, and ill-formed library molecules, which will enable the user to determine the quality of the library construction. This slide is a comparison of balancing 8 differently barcoded libraries loaded at 8 picomolar concentration, which was based on different quantification methods as a function of the percent of the total reads on the y-axis. The 3 different methods compared were ddPCR, the ion equalizer kit, and kappa qPCR. Here, 3 separately pooled sequencing runs were performed, each containing the 8 barcoded libraries at 8 picomolar concentration based on each quantitative method tested. The ddPCR results with the library quantification kit are shown in green. The ion equalizer results are shown in red, and the kappa qPCR results are shown in black. The dotted line at 12.5% is the theoretical balancing percent for perfectly equal representation of each of the 8 libraries in a pooled sequencing run. Clearly, the Bio-Rad ddPCR results are the most consistent of the 3 methods tested, with superior balancing of the libraries. This is the product used for the ddPCR library quantification for ion torrent and all the data that I just showed you. It is available for sale now directly from our website, so please check that out at biorad.com. This is the title of a paper by Lowry et al. 2013 from Biotechniques. Here they designed a probe to Illumina TruSeq libraries in a similar way that we have our Illumina TruSeq library quantification kit. And they also found an inverse relationship between the insert size and fluorescent amplitude, as shown in the lower chart. The larger the insert size, the less efficient the PCR reaction, and thus the lower the fluorescent amplitude of the positive droplets. Here they were testing amplicons ranging in size from 160 to 860 base pairs. This demonstrates that other researchers are taking advantage of the unique properties of ddPCR for library quantification and quality assessment. These are the key takeaway points for ddPCR NGS library quantification kits. They have accurate quantification of the libraries. They provide library quality information, and this is simply not possible with other single technologies. You'd have to combine gels and other techniques to get the same type of information you can get absolute quantification and library quality sense from running a library like that. And this gives you a sense of the library insert size, so a size distribution. I want to introduce the idea that Droplet Digital PCR and NGS platforms are similar in that they are partitioning and counting machines. They achieve partitioning in different ways, as I just showed, but nonetheless, they count DNA molecules. NGS platforms are very good at analysis of a few samples in great sequence detail, whereas Droplet Digital PCR can analyze many samples with one or a few probe assays. The two types of platforms are thus complementary. Droplet Digital PCR can easily be employed to cross-validate NGS sequencing results by probing very low levels of target and also giving very precise quantification of these samples. I will now discuss this complementarity between Droplet Digital PCR and NGS platforms. So how can Droplet Digital PCR be used to cross-validate NGS sequencing results? This is an experiment we performed to compare what happens to the quantification of a known mutant concentration before and after complex NGS sample preparation. Here, we took samples which contain specific ratios of wild-type DNA and mutant for T790M, a mutation in the EGFR kinase. We took these samples and split them into two workflows. In one workflow, we performed Droplet Digital PCR directly on the samples, and the mutant abundance was directly quantitated from an in-house designed T790M TaqMan assay. And this is ddPCR1 shown in green at the top of the workflow. In a parallel workflow, we placed the other half of the sample in NGS library sample preparation for Ion Torrent as well as Illumina TruSeq. The library sample preparation requires many steps with multiple purifications and enzymatic reactions, as well as additional PCR procedures. This complexity of library generation and processing is known to introduce significant biases, and we wanted to see if the ratio of the mutant remains constant through all the processing, or if NGS sample preparation changed the results. Therefore, we sequenced the samples on the respective NGS platforms and also took a second ddPCR measurement after NGS library preparation, and this, in other words, post-NGS library preparation. And this is ddPCR2, and that's shown in red on the lower portion of the workflow. The following tables are the results of comparing ddPCR1, the NGS sequencing results, using the NGS platforms as counting machines to give an answer, independent of ddPCR, and the results of ddPCR2, which is post-sample library construction. This table demonstrates that ddPCR1, which is pre-sample processing, had excellent concordance between the theoretical mutant abundance, which is shown in yellow, for the different mutant ratios to wild-type samples that we tested, and the direct measurement of these samples by Droplet Digital PCR, which is the numbers highlighted in green. Remember, ddPCR1, shown here, is before NGS sample processing. The mutant ratios of the 8 samples tested ranged from 0.02% to 25%. This table is a comparison of the same samples, only now processed through either the Ion Torrent workflow or the Illumina TruSeq workflow. As can be seen, after being processed through the NGS workflows, the ratios of the two NGS platforms, which were determined by counting the reads of the mutant and wild-type sequence, are significantly different from the theoretical percent, which is in yellow. ddPCR2, which is post-sample preparation, and we used the same TaqMan assay as was performed in ddPCR1, which was the pre, was performed on the ion torrent post-NGS sample preparation library. The ddPCR2 results also show significant deviation from the theoretical mutant ratios as denoted by the red percentages. These deviations, given the excellent concordance of the direct measurements of ddPCR1 versus the highly processed samples of the NGS workflows, is likely due to the complex sample processing in the NGS workflows. Thus, ddPCR is a great tool to cross-validate NGS results and is likely a much more accurate quantification method compared to the highly processed NGS library samples. I would like to move on to my final topic, which is using droplets for improved target enrichment for NGS workflows. The overall purpose of target enrichment is to recover amplicons in a high-plexed reaction. For example, suppose a user wants to take 200 primer pairs against 200 different targets, amplify them in one reaction, and sequence them. This is analogous to a custom 200 target cancer panel, for example. Normally, they would mix all 200 primers with template in a single bulk PCR reaction. By using the power of partitioning with droplets, however, they should be able to improve the representation of the 200 amplicons by reducing PCR bias from the many competing reactions. They would add the 200 primers plus fragmented genomic DNA and ddPCR supermix for probes to create a 20 microliter reaction. That reaction is then placed on a DG8 cartridge and used to make droplets in the droplet generator instrument. Then the droplets are transferred to a 96-well plate and thermocycled. After thermocycling, the amplified material is then recovered by breaking open the droplets with chloroform and recovering the upper aqueous phase as shown in the upper right picture. And in the upper right picture, the red arrow on the left is pointing to the chloroform layer, and the red arrow on the right is pointing to the aqueous phase that is formed after a quick centrifugation process. Here's the experimental setup I used to compare a single bulk PCR reaction with a single well of 20,000 droplets starting from the same master mix and template concentration. There are 4 general steps. First, amplify the 200-plex reaction in bulk PCR and in droplet PCR for comparison. 2, recover the amplified 200-plex DNA from droplets by breaking them open. And third, perform standard TruSeq sample preparation, so library construction on both of those types of samples. And finally, fourth, sequence the library on the MySeq sequencer. These are the thermal cycling conditions I used for the custom 200-plex reaction tested. And importantly, in Droplet Digital PCR, we normally use a 2-step, but here we found that a 3-step greatly improved the results. So these are the actual conditions I used in the data I'm about to show you. The 200-plex consisted of amplicons ranging in size from approximately 60 base pairs to 200 base pairs. This is shown in the rank-ordered plot of the 200-plex in terms of amplicon size in base pairs on the y-axis and ranging from shortest to longest on the x-axis. This slide is plotting the same 200-plex in base pairs of amplicon size, the red lines now. However, it also is plotting the percent GC content rank ordered from high to low in blue for each amplicon. Importantly, this shows that the length of the amplicons used are random with respect to the GC content in our custom 200-plex. This slide shows the recovered material from the droplets and bulk reactions run on an Experian gel. The droplet library has a pattern that is more consistent with the predicted sizes of the amplicons from the 200-plex reaction. And the Experian gel, the L is the ladder that was used, D is the material recovered from the droplets, and B is the material recovered from the conventional single tube reaction, bulk PCR reaction that we used for comparison. Sequencing was performed, and the results of the comparison of bulk PCR versus droplets PCR is shown here. This is a logarithmic plot on the y-axis for the number of sequencing reads for each of the 200-plex amplicons sequenced, which are rank ordered on the x-axis from the highest number of reads to the lowest number of reads. The green line is of the reaction performed in droplets, and the red line is of the reaction performed in bulk. Ideally, all amplicons, all 200 of them, would be equally represented, assuming 2 copies of each target per genome. And this is plotted as the horizontal blue line labeled ideal coverage. Clearly, the droplet prepared library in green is more uniform in representation compared to the bulk library. Remember, the y-axis is a logarithmic scale, so the droplet library for many of the amplicons sequenced improved coverage by up to approximately 10-fold in the droplet prepared library. So the conclusions for the target enrichment are: partitioning the samples in droplets reduces competition of a high-ordered multiplex reaction, here a 200-plex, and therefore improves the amplification of the targets compared to the bulk PCR reaction. Better amplification of the targets leads to more uniform coverage in terms of representation in the subsequent sequencing run of the adapted library. And droplet amplification easily integrates into the library construction workflow. So ddPCR is a synergistic technology for NGS. It provides it's really excellent for accurate cross-validation of the NGS results, as I showed you earlier. And you can screen many samples at a lower cost. And so really that's the synergy between digital PCR and next generation sequencing. Library quantification can improve NGS runs by accurately quantifying the adapted libraries for surface loading, and it provides excellent balancing of pooled libraries. This is really difficult to achieve using other technologies such as real-time PCR. Custom amplicon multiplexing can be improved by partitioning in droplets for NGS target enrichment, which I just showed you. And this is the literature available from our website, so please visit the Droplet Digital PCR at Bio-Rad. You can see the website below, www.biorad.com/ddPCR. And we have many manuals, instruction manuals, publication lists, protocols, all available on our website. And I would like to also mention that we are excited to offer EvaGreen Supermix reagent for the QX200 system. This enables a DNA binding dye, EvaGreen, to be used in droplets, which can replace probe assays. EvaGreen works in an analogous manner to cyber type dyes in that it fluoresces brightly when bound to DNA. Therefore, the more DNA that is amplified, the brighter the signal in the droplet. And so this replaces TaqMan type probes. So more information can be found on our website at biorad.com. And I just want to thank you. This photo is of the ribbon cutting ceremony for the new Digital Biology Center in Pleasanton. And there's my contact information. And again, these slides will be available for you to look at later. They'll be posted. So now I will turn to questions and answers.
Thank you, Nick, for that very engaging talk. If you would like a copy of today's PowerPoint presentation, a copy of the QX200 ddPCR and Digital PCR Assays brochure, and more literature on the Ion Torrent and Illumina Quantification Kits, they are available at the link to the left of your screen for download. The webinar is also available on demand within 24 hours. If you would like any additional information, please feel free to reach out to your local Bio-Rad Droplet Digital PCR specialist as well. Nick will now take questions. Nick, your first question is, can ddPCR detect one copy?
Yes, it can, actually. ddPCR is extremely sensitive. It's orders of magnitude more sensitive than qPCR. And so ddPCR can easily detect a single copy.
Along the same lines, the second question is, how feasible is this method for very rare sequence detection like heterogeneous mixtures such as tumors or mixed microbial populations?
So if I understand the question correctly, like using ddPCR as a synergistic tool to detect that?
That's correct.
Yeah, so I think ddPCR is excellent for that. You have to design assays to those targets, but your quantification will be exceptionally good with digital PCR compared to your NGS sequencing.
Speaking of NGS sequencing, are the next generation sequencing results always wrong compared to droplet digital PCR?
That's a great question. No, it depends, I think, on many factors. Probably likely which kits were used in the library construction. I guess I would say specifically how much processing was implemented and what levels of accuracy are important to the user. So this is why ddPCR is synergistic with NGS in the cross-validation. I know there's some newer kits that have less processing, but a lot of people still go. It takes about a day and a half, 2 days to generate their libraries before they're ready to do sequencing. So all that processing, all that PCR, all that purification, bead cleanup can actually bias your results, and I think that's where ddPCR can help give you confidence if the questions you're asking are quantitative.
So our next question is from a user. Do you have any best practices for making the serial dilutions or while prepping droplet digital PCR reactions? Yes,
that's a great question. So typically we do our dilutions on a 96-well plate format. You can use water or a low TE buffer. You don't want to have too much EDTA that might inhibit PCR and could affect your results. So just a low TE buffer you can buy. That's great. I do recommend doing all your mixing on the 96-well plate. You can pipette up and down, say like 10 times to make sure that the dilutions are mixed very well before you go on to your next dilution. And never mix your ddPCR reactions on the DG8 chip. You should always mix them in tubes or on 96 well plates, because once you start loading the DG8 chip, due to the microfluidics on the chip, it starts drawing the fluids onto the chip immediately. And so even though you might think you have time, you probably won't have time to mix properly on a chip. So never mix on a chip.
So during your talk, you mentioned extracting amplified products or breaking open the droplets. Is there a protocol available for this, and how long does it take?
Yeah, that's a great question. So we have all the kits come with a master mix, and that master mix does not have DUTP in it, so it has the native nucleotide. So the master mix is called Droplet PCR Supermix for probes, no DUTP, and that's really important to use in all these applications, to use the native DNTPs. So in order to break open the droplets, there's a protocol in the product insert that comes with that master mix. But briefly, it's you transfer your droplets to like an Eppendorf tube. So you transfer everything, the oil and the droplets from the well, into an Eppendorf tube. You can combine replicates if you want. You remove the oil, you add chloroform, you vortex, and then you're going to spin at, I believe, 15,000 G, and then for about 10 minutes, and then you'll get the separation of the broken droplets. So you'll have the chloroform on the bottom and the upper aqueous, which has your recovered amplicons from the droplets. So very easy, takes about 15 minutes. And you can parallelize that if you want.
So Nick, towards the end of the talk, you mentioned EvaGreen. How do you exclude non-specific amplification when using EvaGreen with Droplet Digital PCR?
That is a great question, and I wish I had more time to talk about EvaGreen. My talk is on NGS, but briefly, because of the properties of the amplitude fluorescence, which I showed you with probes, EvaGreen also has some very unique characteristics that are very exciting. So with EvaGreen, the smaller the PCR product being amplified, the less bright it is, which is inverse to what I showed you with the probe-based. So with like adapter or primer-dimer species, it'll be a very low fluorescent positive droplet, and then your true positives will be very high fluorescent because those are longer in base pairs. And so you can actually distinguish them on the 1 and 2D plots quite easily. And using our thresholding tools and lasso tools in Quantisoft software, you can actually get accurate quantification, and you can actually quantitate primer-dimer if you wanted to, something you cannot do in real-time PCR. Very exciting.
So along the same lines, we have a question regarding Droplet Digital PCR and multiplexing. Is it possible to multiplex using Droplet Digital PCR, and how many targets can you multiplex?
Again, that is a great question and probably a whole another hour talk. But briefly, yes, we have done multiplexing, and many people out in the field, the hundreds of ddPCR users out there are playing with multiplexing. There's many ways to do it. You can vary the probe concentrations. You could vary temperatures. You can vary the primer concentrations. And basically what happens is when you do that, you are shifting the clusters in the 2D plots. And so there's a whole space there that you can spatially separate your clusters by fluorescent amplitude. And by playing with those parameters, you can actually shift the clusters, and you can add more clusters. And so with our lasso tool, you can quantitate those clusters. And currently, I think in-house, we typically have done about probably about 4 to 6 different targets in multiplexing, but probably more is possible. It just is going to take a little fine-tuning, a little upfront work. But yes, you can absolutely multiplex in the Droplet Digital PCR system.
Our next question is specific to the library quantification kits. There are numerous serial dilutions that are needed before quantification. So is there a way to simplify this process?
Currently, this is our best recommendation, is to do the serial dilutions. It is analogous to the serial dilutions that you have to do in qPCR. But again, remember, you don't have to load a standard curve on your plate. So you actually have more wells potentially to play with on your plate if you're doing a lot of samples. And with ddPCR, you can really get away with one sample if you want. We do advise replicates just in case a mistake is made or something happens, but generally one well, because you have roughly 20,000 droplets are generated per sample, so you have plenty of measurements per well to get an accurate quantification. But in terms of getting away from the serial dilutions right now, no, you still have to do the serial dilutions.
Are there any ddPCR diagnostic kits available now or in the future?
So I'm not quite sure I understand for what application the diagnostic kits. We do have rare mutation assays, and we have a whole slew of assays available on our Prime PCR for copy number variation. So we are offering content on our website, so I do recommend you can check that out.
Can we use the currently available Prime PCR digital assays for validation of the NGS data?
So as long as the Prime PCR copy number assays are within your library that you made, or you could add them in if you wanted to, you can add those targets in, then yes, you can absolutely do that. And that would serve as a great reference to verify that you can run ddPCR like I did, and then in parallel do the sequencing and let the sequencer tell you what the answer is. And you can see if there was any biases in your sequencing results quantitatively. So yes, you can. And of course, as I just mentioned, we have many rare mutation assays coming online that we sell through our website for ddPCR.
So you mentioned qPCR earlier, and that serial dilutions are also required for qPCR. Then why is ddPCR better than qPCR for library quantification?
Yeah, so again, with qPCR, you're reliant upon the standard curve. You probably have to do triplicates, and many times you throw out one of your triplicates because it's the CT is significantly different from the replicates, and so now you have to go in and kind of selectively decide which well to throw out on your triplicate results, and so you're only getting really 3 measurements on qPCR, whereas in digital PCR, you're getting tens of thousands of measurements in one well, and if you did duplicates, you're doubling that. So really, that's how we get our extremely high accurate results, and that's why, as I showed you with the ion torrent workflow, I showed ddPCR results using our library quantification kit and got excellent balancing of the 8 libraries. With the ion equalizer kit, which is a bead-based exclusion type method, it's basically based on the capacity of the beads to bind the DNA to a certain concentration. Those results were, off by like up to, 150, 200% variation there. And with the qPCR results, which is what people typically do, again, there was a huge variation. Let me see if I can push to that slide so we can show that again. That was very, very exciting data. Let me just try and bring that up. There we go. So again, here's the results comparing qPCR in black and ddPCR in green. And again, the dotted line, the 12.5, is the ideal balancing of the libraries, the 8 libraries. Remember that the green, all 8 are a single run. The red for ionic equalizer, all those are a separate run, and the black is a separate run for the qPCR. So again, the qPCR results are they're just not as good, and that's because you just don't have the accuracy and precision that digital PCR offers.
Thank you, Nick. That was our last question. And thank you, everyone, for attending the 4th Bio-Rad Droplet Digital PCR webinar today. We have 2 more webinars in the coming months, so please participate in the full webinar series. As a reminder, today's webinar will also be available on demand within the next 24 hours. For those of you who had questions we were not able to get to today, someone from Bio-Rad will follow up with you in the next few weeks. For more information on Droplet Digital PCR and educational resources, visit www.biorad.com/webinar. This URL is displayed to the left of your slide in the resources box. Thank you again.